Hello, I'm Robin Puglia, and this is the Cyrex RA2 educational pathway. In this video, we'll be exploring some of the details of intestinal permeability. So, when we refer to leaky gut, or even to intestinal permeability, what we actually mean is inappropriate or pathogenic intestinal permeability, because intestinal permeability is actually an intrinsic property of the intestine. So permeability refers to the process by which the intestinal epithelia, that single monolayer of cells, allows molecules to pass through, and it refers to the passage of ions and to low molecular weight molecules. So this is in contrast to transport, or to intestinal transport, which relates to the number of molecules crossing the epithelial layer at any given time. So this is important to understand because Cyrex is the only lab that really considers the immunogenicity of permeability. So it is less about the passage across the wall and more about whether or not there are immune and inflammatory consequences to the passage of molecules, or whether permeability or passage is due to damage or injury to the gut itself. And again, this is really important to clinical practice because these factors will, or certainly should, greatly influence the interventions or the further investigations that are needed. So if there is damage to the gut, the outcome is vastly different to low-level inflammation or just passage of small molecules with no immune implications. So a common reason that I hear from practitioners regarding why they don't test for gut permeability is adverse food reactions. So the thinking being, that if somebody has food sensitivity or food reactions, then this must equal leaky gut, and so no need to test, that the food reactivity test itself is an intestinal permeability test. But in fact, there are multiple mechanisms of dietary protein transport across an intact barrier. And these include dendritic sampling. And the image that I've included here, you can see that the dendritic cell, in fact, can reach up through the epithelial cells to sample proteins from inside the gut lumen. And the image of this instance is a bacteria, but this also happens with food protein, dietary protein antigens. Transcytosis mechanisms, immunoglobulin-mediated transport, and a mechanism which is called ectopic expression of CD71, which I'll demonstrate in one of the slides following this one. So in celiac disease, for example, enhanced permeability to gliadin peptides can actually result from transport from the apical part of the cell to the basal part of the cell, via secretory IgA binding onto the CD71 receptor on the apical surface of the cell. So that's one fairly significant example of food protein being transported across an intact gut wall that has significant inflammatory and immune consequences without there needing to be permeability present first. So we can't really talk about permeability without, of course, talking about the junctions and the junction proteins. So there are a large set of structural and regulatory molecules that control the plasticity and the permeability of the tight junctions, and these are mainly comprised of proteins. So these include the claudins and occludins. They include junctional adhesion molecules that you see here, and other proteins. So occludins, similar to claudins, interact directly with the membrane-associated zonular occludins, so this little ZO proteins that you see over here. And the entire set of proteins communicate together under the influence of zonulin, the enzyme zonulin, to influence whether or not the tight junctions remain tightly closed or become more open in order to facilitate transport of molecules through that tight junction there. So healthy paracellular transport includes the normal passage of molecules between the adjacent epithelial cells. So this process is part of a healthy, well-regulated gut function. The tight junctions act as a rate-limiting step, which is as it should be, and it is passive. The transport, paracellular transport, is passive, so it depends on concentration gradients, and it also is a process independent of tight junction regulation that can affect mucosal permeability. Transcellular transport, though, is the pathway of substances taken through the intestinal epithelial cells via transcytosis. So this can happen, of course, as part of a healthy functioning gut wall, and it is one of the mechanisms through which dietary food proteins can cross into the systemic system in the absence of permeability. And we see here that transcellular penetration of antigens, of course, is happening through the cell itself. However, when we see transcellular hyperpermeability, so when this mechanism in fact has gone awry and has become pathogenic, it is not part of healthy cell transport. There are 3 reasons that we see markers such as actomyosin antibodies elevated, which would indicate transcellular hyperpermeability, and those include severe significant inflammation such as undiagnosed intestinal autoimmunity or undiagnosed celiac disease, or they can also be indicative of intestinal infection where the cell wall is actually becoming damaged. So in this instance, we're not talking about garden variety intestinal permeability, where if you use a little bit of glutamine and take out some of the common allergens, the gut wall is going to repair. There is a significant amount of inflammation, a significant amount of oxidative stress, and we're talking about injury to the gut wall in this instance. So the clinical outcome and your clinical decision making needs to be different if you have transcellular hyperpermeability versus paracellular hyperpermeability, where the clinical thinking and the protocols should be quite different. Okay, so exosomes and food-derived antigens. So the high concentration of foreign antigens that are present in the gut lumen are separated, of course, from the intestinal immune system via the gut wall. And intestinal epithelial cells can endocytose dietary proteins and process them into peptides. And in vitro, they can actually present these peptides to CD4 positive T lymphocytes. And we see here the CD4 positive receptor on the T cell in conjunction with the antigen presenting cell with MHC class 2. So the major histocompatibility complex class 2. When you have this interaction, then we have the ability to call into action or to upregulate Th1 cells, Th2 cells, and Th17 cells. So we see antigen presentation that directly calls into play the pro-inflammatory immune response. So this means that you can have a degree of food reactivity without, like I said, classically defined intestinal hyperpermeability. And again, it means that if you have a food reactivity test that comes back, then this does not necessarily mean that the person has, classically defined intestinal permeability. So you need to really be running an accurate test to look at the permeability itself and to understand if you're working with an immune-mediated issue. So you need a test that really helps you understand the immunogenicity of the permeability, which is to say that you need to be running Cyrex Array2 because, of course, this is the only intestinal permeability test available on the market that actually does that looks at the immune consequences of the permeability. So I wanted to go a little bit more into antibody-mediated transport. So we're going to start with secretory IgA. So of course, secretory IgA is a very, very important part of intestinal protection and protective immunity in the intestine. And it does that by retaining potentially problematic antigens or dealing with potentially problematic antigens in the lumen of the gut. in order that it doesn't cross into and require the intervention of the systemic immune system. So again, celiac disease is a gluten-reactive enteropathy that is induced by abnormal activation of the T cells by gluten-derived gliadin peptides. So the breakdown proteins from gluten, one of which includes gliadin. So in celiac disease, though, gliadin peptides are transported intact across the intestine through an IgA-mediated transport process. So you'll see in this slide here, you can see the image. And here we have this receptor, which I referred to before, which is CD71. So we often see the receptor expressed here on the basement membrane part of the cell. But when you have expression or overexpression of the CD71 membrane here on the apical part of the cell, then we have the ability to create IgA-mediated transport via the CD71 receptor, where the IgA bound to food protein is able to actually cross directly through the cell itself into the lamina propria, where the food protein will come into contact with the systemic immune system, at which point various different types of inflammatory mechanism can be activated, including but not limited to the autoimmune process itself, as we would see in celiac disease. So secretory IgAs are mainly devoted to restricting the entry of antigens. And in some cases, this apical to basal transport using the IgA mediated transport can be protective. So you'll see on the left-hand side of the slide here, you see that with Shigella infection, that IgA-mediated transport, in fact, allows the infection to be taken care of by the systemic immune system, and this actually helps to preserve the intestinal barrier integrity. So the body does that, and it's a protective mechanism in order that the gut doesn't become damaged by the infection. So we see that this mechanism can be beneficial because, of course, the body doesn't do anything for no reason. So it can be beneficial and protective, and it can also be problematic and upregulate or directly contribute to autoimmune disease. So then we have a similar mechanism involved with IgE-mediated transport using a different receptor. So the CD23 receptor can be used to help transport IgE-mediated antigens from the gut lumen into the lamina propria. And in this instance, allergens complex with IgE bypass epithelial lysosomal degradation. So they're not broken down. And this, of course, creates a much larger target for immune recognition and immune activation. And with IgE-mediated responses, of course, we're talking about mast cell activation and classical allergy symptoms as a result of that. Okay, so IgG-mediated transport. So IgA, of course, constitutes the predominant type of immunoglobulin at the mucosal surfaces. But actually, gastrointestinal secretions contain a significant amount of IgG also. And conventionally, it is really considered that IgG can be considered protective. And part of that thought process is because IgG, of course, is the immunoglobulin that helps to protect a newborn infant that is transferred from the mother to the infant. So this IgG-mediated intestinal transport seems to really be implicated in protective immunity. It helps us to tolerate, in fact, dietary antigens, and this is especially recognized in the context of consumption of IgG via breast milk. So here we have again, when you have IgE-mediated transport through the receptor on the apical part of the membrane here, the ability then, it bypasses degradation here in the interior of the cell, and the food protein is actually presented directly to mast cells, initiating inflammatory mediators, resulting in classical allergic symptoms. So this mechanism, of course, is considered to be significantly upregulated in dietary food protein allergy, classical allergy. And then we also see on the left-hand side of the slide the very similar mechanism using IgG, which is thought to be a protective. When this occurs in IgG, it's thought to be protective. So to summarize, there are multiple mechanisms via which food proteins can cross an intact gut wall and to come into contact with the systemic immune system. So if you have somebody with multiple food reactions, testing to rule in or rule out intestinal permeability allows you to create targeted and specific interventions. If there is not inappropriate permeability involved, then you do not need to waste time and money on protocols and nutraceuticals that are targeted at repairing permeability. If you go down this path because of an assumption that you made clinically, then you might find yourself 3 months or 6 months later without actually having made progress and not understanding why, or not understanding how to proceed forwards from there. You may also have missed the opportunity to really understand the mechanism behind loss of dietary protein tolerance because you haven't had the opportunity to investigate it further, or even potentially missed an undiagnosed or gut-related autoimmunity. So in our next video, I'm going to be discussing paracellular versus transcellular pathogenic permeability in more detail. I hope you'll join me.